Hello dear students, in this video lecture you will have a short review of the basic concepts of DNA and RNA. You have already learned these topics in detail in your cytogenetics class, so let us refresh your memory with these essential topics. At the end of this short video lecture, you are expected to demonstrate the following. First, correctly recall the difference between DNA and RNA. Describe correctly the functions of the two types of nucleic acids, and lastly, to correctly identify and describe the ways by which mutation happens. At this point, let us talk about the DNA or deoxyribonucleic acid, its historical background, and its composition. When DNA was not yet discovered, scientists believed that proteins bear the genetic material of cells. DNA was first isolated in 1869 by Friedrich Nischer from Switzerland, who discovered microscopic substances from pus among surgical bandages. Because it was found in the nucleus of the cell, it was first termed as nuclein. At this time, proteins were generally accepted as the hereditary material. DNA was not known to carry genetic information until 1952 when Alfred Hershey and Martha Chase performed a series of experiments on the T2 phage, which is a type of virus that eats bacteria. The Hershey-Chase experiment provided concrete evidence that genes were made up of DNA. The Hershey-Chase experiments settled the long-standing debate about the composition of genes, thereby allowing scientists to investigate the molecular mechanisms by which genes function in organisms. In 1953, Francis Crick and James Watson introduced the currently accepted model of the DNA, that of the double helix, which led them to receive the Nobel Prize for Medicine or Physiology in the same year. Now, DNA or deoxyribonucleic acid is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. Most DNA is located in the cell's nucleus, where it is called nuclear DNA, but a small amount of DNA can also be found in the mitochondria. DNA molecules are made up of nucleotides. A single nucleotide is made up of 3 parts: a phosphate group, a 5-carbon sugar, and a nitrogenous base. For DNA, the sugar attached to the phosphate group is deoxyribose. Nucleotides are joined together through a covalent bond between the sugar group of one nucleotide and the phosphate group of the next in a sugar-phosphate alternate backbone. The information in DNA is stored as a code made up of 4 chemical bases or nitrogenous bases. 2 are purines and 2 are pyrimidines. These are adenine, guanine, cytosine, and thymine. Human DNA consists of about 3 billion bases, and more than 99% of those bases are the same in all people. The order or sequence of these bases determines the information available for building and maintaining an organism, similar to the way in which letters of the alphabet appear in a certain order to form words and sentences. DNA bases pair up with each other, adenine with thymine and cytosine with guanine, to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Nucleotides are arranged in 2 long strands that form a spiral called double helix. The structure of the double helix is somewhat like a ladder, with the base pairs forming the ladder's steps and the sugar and phosphate molecules forming the vertical side pieces of the ladder. So now let us talk about the RNA, or the ribonucleic acid. Like DNA, the ribonucleic acid is an important molecule in the regulation and expression of genes. RNA transfers the genetic code from the nucleus to the ribosomes during protein synthesis. Aside from its important roles in protein synthesis, RNA also has other biological functionality, such as communicating cellular signals, as well as catalyzing some chemical reactions. RNA is found to be single-stranded with ribose as the sugar backbone. With it are the nucleobases adenine, guanine, cytosine, and uracil. Uracil serves as an alternative for thymine as a nucleotide base. However, it is just an unmethylated form of thymine. We have this figure to differentiate DNA from RNA. Ribonucleic acid, or RNA, is a molecule similar to DNA. However, unlike DNA, RNA is a single-stranded molecule. RNA is chemically similar to DNA except that it contains ribose instead of deoxyribose as its sugar and has the nitrogenous base uracil rather than thymine. Thus, each nucleotide along a DNA strand has adenine, guanine, cytosine, or thymine as its base, and each nucleotide along an RNA strand has adenine, guanine, cytosine, or uracil as its base. Different types of RNA exist in the cell. Of the many types of RNA, the three most well-known and most commonly studied are messenger RNA, transfer RNA, and ribosomal RNA, which are present in all organisms. The first type of RNA is the messenger RNA, or mRNA. mRNA is a single-stranded RNA molecule that is complementary to one of the DNA strands of a gene. Since DNA is too large to successfully exit the cell's nucleus, the genetic information is carried by the messenger RNA to the cytoplasm. The mRNA, which is a product of the transcription process, is an RNA version of the gene that leaves the cell nucleus and moves to the cytoplasm where proteins are made. Next is the transfer RNA, or tRNA. tRNAs are small RNAs with a size of about 80 nucleotides. Its main function is to transfer amino acids to growing polypeptide chains at the ribosomal site of protein synthesis. Each tRNA molecule has two important areas: a trinucleotide region called the anticodon and a region for attaching a specific amino acid. Lastly is the ribosomal RNA, or rRNA. rRNAs comprise the majority of RNA found among typical eukaryotic cells. These are the catalytic components of the ribosome. rRNA and proteins combine to form the ribosome, which later on binds with mRNA for protein synthesis. As a summary, this slide explains the relationship of DNA and RNA. Genes provide the instructions for making specific proteins, but a gene does not build a protein directly. The bridge between DNA and protein synthesis is the nucleic acid RNA. This concept was dubbed as the central dogma by Francis Crick in 1956. The central dogma of molecular biology describes the flow of genetic information in cells from DNA to messenger RNA and then to protein. It states that genes specify the sequence of mRNA molecules, which in turn specify the sequence of proteins. Because the information stored in DNA is so central to cellular function, the cell keeps the DNA protected and transcribes it in the form of RNA. An enzyme adds one nucleotide to the mRNA strand for every nucleotide it reads in the DNA strand. The translation of this information to a protein is more complex because 3 mRNA nucleotides correspond to one amino acid in the polypeptide sequence. This is the end of this short video lecture. Thank you for listening.